Hello, my name is Catherine Inman, and I am creating this video for my cell biology course at Sam Houston State University. The protein I'd like to discuss today will be DNA polymers. We'll look at a couple images and go over their functions in the cell. First, we'll look at the image over here. It is the fingers over there. No, wait, that way. Okay, over that way. It's going to have fingers, thumbs, and palms. And the DNA polymers is going to more act like a right hand. And so that's why it's given some of those names. The fingers are involved in the interactions between the nucleoside triphosphate being inserted and the existing template base. The thumb acts more to assist the aligning of the double-stranded DNA. Finally, coming to our palm, catalyzing the transfer of phosphoryl groups in the reaction, which is catalyzing the essential amino acids in those active sites. So that's really cool, and I have had a super fun time learning about this protein. I could make this video go on a little bit longer, but I feel like I have answered the questions, so Miss Dr. Hudson, if you would like to stop here, you can. For anyone else still interested in listening, I would like to go into the building blocks, the template blueprints, the primers, and other things that the DNA protein might need for educational purposes. So, in the other picture I have selected, I will move my screen a little bit smaller. Okay, it's not working. I'm very new to making videos, so we'll just make it a big screen. All right, DNA polymers. So they're really super interesting, and you see right here, this is where the DNA polymers takes place, and it takes place in double helixes forms. These are our leading strands, our primers, which are needed in order to get the DNA polymers to happen. And I'll just jump right into it. So there's three things that you absolutely need in order for the DNA polymers to happen. Those are the building blocks. We call them the bricks or the DNTP molecules. And that stands for a really long word that I have. Okay. The dioxynucleoside triphosphate. DNTP. And there's 4 different types of them. There's dioxydoensine, 5' triphosphate, dioxoguanosine, 5' phosphate, dioxocytine, 5' triphosphate, and finally the thymidine, 5' triphosphate. I don't know if I'm pronouncing those right, and maybe I could even type in the correct spelling of them in the description below. The next thing that we're going to be needing are what we can call like the blueprint or the instructions, which is the DNA template strand that the DNA polymer can only add new nucleotides to. And without the blueprint, they're just not going to be able to extend and build that chain. Finally, we need the primer to initiate the replication, and this is where the first bond arises from. The sequence of the nucleotides that are already attached to the DNA template. You'll also need magnesium ions, which increase the efficiency of DNA polymers, and it's just really interesting to know that in 100 million, 100 million correct attachments that the DNA polymer makes, they only make one mistake. So the last thing that's not necessary to have, but that the DNA polymers obtain or possess, is the ability to correct their own mistakes. Any mismatches in their base, which it really does make a mistake, like I did say, 1 in every 100 million. So that's pretty cool. It's also interesting to know that the DNA polymers can only move from the 5 to the 3. They cannot move from the 3 to the 5, and that is because of the anti-parallel direction in which separate to synthesize. Back in Uh, the other photo, it was hard for me to determine which one of those ribbons are the primary, secondary, tertiary, and the quad. I know that in order to have a quad, it refers to the 3D interactions between multi-sunbelts, and I don't know how many sunbelts there are. That's still something I'm learning. If you know, please comment in the section below, or Dr. Hudson, when you're watching this, if you could reach out to me, or maybe I'll send you a private email. Not to make this video so long, but the primary structure has to do with the linear sequences of the nucleotides. So I would just imagine that that would be taking place in the part A, the building blocks and bricks. And to me, it looks like that's going to be those green structures. Then finally, the secondary, the small local folding TIFs, I would imagine those would be the pink ones. And the tertiary, the 3D folded shape of the nucleotide molecule. And I just hope those are the yellow ones. I'm trying to point out which ones they are. I'm once again not sure, but I hope that I was able to educate anyone watching this video on DNA polymers. Thank you for watching. And everybody always says like and subscribe, so if you want.